This lesson's on enzyme inhibition, and here are your objectives. First, just a reminder that metabolic pathways are arranged in chains and cycles. Here I'm indicating several compounds, each being converted into the next, and each arrow represents an enzyme catalyzed reaction. I'm indicating those with numbers. We could also have those arranged in a cycle, as we'll see when we get to photosynthesis and cellular respiration and the Calvin and Krebs cycle. And there could be things feeding into and coming off of each of these reactions. Again, each reaction would be enzyme catalyzed. And these can be, as I've shown in the cycle, branched. I'll show that here in the pathway as well. And that allows us to make several different products from one starting point. This kind of setup allows an exquisite degree of control because, for example, a product at the end of a pathway could actually feed back and inhibit an enzyme earlier in the pathway. That's what I'm showing here. So as D builds up, it actually comes back and switches off or turns the volume down on enzyme 1. And that would slow production of D itself. This is a clever way of regulating the amount of D that's produced. As more D is made, it switches off enzyme 1. As less D is made, there's less to switch off enzyme 1, and so it's self-regulating. This is called end product inhibition and is an example of feedback inhibition. Feedback inhibition overall, or negative feedback, is a key process that we'll see again and again in biology. And a specific example of this is the conversion of the amino acid threonine into another amino acid called isoleucine. As isoleucine builds up, it inhibits the first enzyme in the pathway called threonine deaminase and turns the volume down on it, makes it less effective, and so decreases the production of isoleucine itself. Again, end product inhibition. Now, that typically occurs on the first enzyme in the pathway because that's the most efficient way of doing it. It prevents the buildup of intermediaries if you inhibit the first enzyme in the pathway. If isoleucine were to inhibit this enzyme here, you'd get the buildup of B and C, which might be wasteful. So end product inhibition tends to act on the first enzyme in a pathway. Let's talk a little bit more about enzyme inhibition. Broadly speaking, we can categorize enzyme inhibition into competitive and non-competitive. In competitive inhibition, the inhibitor or the inhibitor molecule resembles the substrate and actually competes for its spot in the active site of the enzyme. So let's say that the blue Pac-Man is my enzyme, the green pie is my substrate, but the red here, which resembles the substrate, is my inhibitor. And because it resembles the substrate, it can bind to the active site and just block the substrate from getting access to it, and therefore slows the rate of reaction. So competitive inhibitors resemble the substrate and compete for access to the active site. Remember that enzyme catalyzed reactions are all about collisions, so these things are jumping on and jumping off of the active sites very, very quickly. And so if you have lots of inhibitor around, it will tend to crowd out the substrate. Non-competitive inhibitors, in contrast, don't resemble the substrate, and they actually bind to another spot somewhere on the enzyme. So here is an example of a non-competitive inhibitor, and what it's going to do is it's going to change the shape of the enzyme and the active site, and now the substrate can no longer bind. So the substrate now can't bind because the active site has been changed in shape by the non-competitive inhibitor binding somewhere else. Related to this is the concept of allosteric regulators, and they can be inhibitors, but they can also be activators. In general, allosteric regulators are things that bind to proteins and change their overall shape, so that they are either more, in the case of activators, or less, in the case of inhibitors, active. So these two terms will get used somewhat interchangeably at this level, although if you study enzyme kinetics in a lot more detail, you'll differentiate the two. And in fact, we'll see an important example of allosteric regulation with hemoglobin. Now, hemoglobin is not an enzyme. It's a transport molecule for oxygen in the blood, but it changes shape when it gets bound by oxygen, and we will see that that's important in its function. An example of a non-competitive inhibitor that we've already seen in this lesson is isoleucine. Isoleucine inhibits the first enzyme in the pathway leading to its own production, and it does this by this kind of non-competitive or allosteric mechanism. An example of a competitive inhibitor are the statin drugs, or HMG-CoA reductase inhibitors. Now, HMG-CoA reductase is an enzyme that's important in the synthesis of cholesterol in the liver, and statins, drugs like Lipitor that you may have heard of, act as competitive inhibitors of that enzyme, and therefore they decrease cholesterol synthesis. And so they are prescribed for people who have high blood cholesterol to help decrease their cholesterol and their risk of heart attacks or strokes. In general, enzyme inhibition is an important mechanism of action for many drugs. Let's finish up by talking about how you can distinguish these different kinds of inhibitors on a graph of rate of reaction versus substrate concentration. First, just a reminder that as you increase substrate concentration, you see an increase in the rate of reaction until ultimately you see a plateau as the enzyme becomes saturated. Now, if you have a competitive inhibitor, this is my enzyme, and I've got substrate out here. A competitive inhibitor is competing for the active site on the enzyme. And so if these are competitive inhibitors, this is going to lower the rate of reaction because I now have a pretty good chance of having a competitive inhibitor get access to the active site instead of a substrate. But if I start increasing the concentration of substrate without increasing the concentration of inhibitor, now it's much more likely that I'm going to get a substrate onto the active site and therefore a reaction than an inhibitor. So increasing the substrate concentration, I can outcompete the competitive inhibitor right back. So what that means on a graph of rate of reaction versus substrate concentration is that you see something like this: lower rates of reaction than the uninhibited state that I've drawn in black, but as you increase the substrate concentration, you can ultimately get back up to the same rate of reaction because you're outcompeting the competitive inhibitor right back. So this is competitive inhibition. In contrast, if you have a non-competitive inhibitor is going to bind to the enzyme and take it out of commission. And so it doesn't matter if you start trying to swamp it with more substrate, because the enzyme is effectively poisoned from elsewhere, you can't bring that rate of reaction back up. So a non-competitive inhibitor has a rate of reaction versus substrate concentration graph that looks like this. Increasing the substrate concentration never gets you back up to the regular rate of reaction of the non-inhibited situation because you've effectively poisoned the enzyme. So a non-competitive inhibitor looks like this. Again, the difference is with a competitive inhibitor, increasing the substrate concentration, moving rightwards on the graph, you can outcompete the competitive inhibitor. You can't do that with a non-competitive inhibitor because it's just decreased the effectiveness of each and every enzyme. So that's a look at enzyme inhibition.